Hello, thank you for tuning into my talk. My name is Nicholas Izzo. I'm Director of Pharmacology at Cognition Therapeutics, and I'm going to be talking about context-dependent pharmacology of sigma-2 receptors in neurons. I am a full-time employee of Cognition Therapeutics, where I am Director of Pharmacology. I'd like to acknowledge our generous support that we've got for a lot of these studies over the years. I'd like to remind people also that the information in this presentation may not be shared on other platforms. The main questions I'm talking about is how we define the pharmacology of the sigma-2 receptor. People who work with this, as all of you probably do, have heard, are often asked whether a particular sigma-2 ligand is an agonist or an antagonist. I think we need to be moving away from these blanket terms. It's a much more complicated picture with this protein receptor. Due to the number of different functional outputs of the receptor and different protein interactions that are involved with its function. And I think that we should start looking at the sigma-2 ligand functionality as being dependent on the biological context. If we look at what the definition of a receptor is, you have to remember it's not just a binding site, this is a functional unit. So for sigma-2, we have cellular macromolecule, in this case, TMEM97, PGRMC1, and possibly other proteins that have not yet been defined. We have a drug interacting site, which is the binding site on the TMEM97 protein. And we've seen some evidence presented at this symposium in the past that showed that this binding may be influenced by the presence of PGRMC1, possibly other proteins. Then we have a cellular or systemic response, and this is where the picture gets a lot more complicated. I'm going to be talking about the effect of sigma-2 ligands to counter the downstream effects of beta amyloid oligomers in neurons. But we'll also hear talks about several other functional consequences of the sigma-2 receptors. What we won't hear about today, but I'd like to draw your attention to, is some additional work that we've found looking at sigma-2 receptors and their effects to mitigate the downstream effects of alpha-synuclein oligomers. This work has been recently accepted for publication. You should be seeing that in press. So with this wide spread of functional consequences of sigma-2 receptors and ligands, is the pharmacology of ligands the same in every case, or is the pharmacology context dependent? So like I said, I'm concentrating on showing you the functional consequences and pharmacology of sigma-2 ligands versus A-beta oligomers in neurons. And what we've used is a vesicle trafficking system in which a cargo dye is added into cultures of neurons. It's taken up into the neurons, into the cells, and is converted to a dark substrate, which is packaged up into vesicles and exocytosed from the cells, where it is crystallized as the dye hits the extracellular media. The exocytosis process is sped up by the addition of A-beta oligomers. And what you see here then is looking at a single time point where you have a vehicle up at 100%. When you add A-beta oligomers, you end up with less dye remaining in the vesicles because it's been turned into crystals. Now, if you look at sigma-2 compounds in here, we see a number of different functional phenotypes. The first phenotype is exemplified here by the sigma-2 ligand SM21. Now you can see when this compound is added to this cell system, you see a dose-dependent reversal of the A-beta effect on vesicle transport. So these types of responses we refer to as an A-beta oligomer antagonist response. There have also been compounds characterized as sigma-2 agonists because of their cytotoxic effects. What we found in this system is that when these compounds, and in this case you're seeing the results of ceramicin, what we see as concentrations of this drug increase, you start seeing a reversal of the A-beta effects on trafficking at lower concentrations, but at higher concentrations, you see this crash of the signal. And this crash is actually due to cell death, which we can see in the panel on the right, where the dye has not been actually taken up and converted into the dark substrate. We also have seen a number of other compounds, sigma-2 compounds, such as this seriniac compound, which are inactive. So here, up to the highest concentrations we've tested, we don't see any effect on the A beta. So these are just referred to as inactive compounds. We have characterized a number of published sigma-2 ligands that I'm sure most of you are familiar with. And what we have found is that there are a subset of the compounds that do have this anti-A beta effect. And you can see 4 of them represented here. These are all low nanomolar affinity compounds at the sigma-2 receptor, and these all have anti-A beta effects. We have also found other sigma-2 ligands that are inactive, in addition to the ones I showed you. And interestingly, DTG, which is the canonical radioligand for the sigma-2 receptor, is inactive in this assay. Not sigma-2 receptors, but I just want to show you progesterone and the progesterone antagonists. Neither of those are effective in this assay. So these are very potent compounds that part of perhaps related to the complex that don't have an effect and are not sigma-2 ligands themselves. And then we have sigma-2 agonists, which I've shown you before, have an anti-A beta effect in every case, and then a cytotoxic effect at higher concentrations, which is associated with activation of caspases. So the question still remains of whether this toxic effect at higher concentrations is mediated through the same pathway that this anti-A beta effect is, or whether there's another receptor or just another pharmacology that's being enacted by a different modulation of that sigma-2 binding site. Now, situation's just getting complicated because we're looking at different types of binding events. First, we're talking about competitive inhibition at the sigma-2 binding site, and this can be demonstrated here, where, as you've seen, you have some inactive compounds like 3-O-ifenpridil, and then you have active compounds. These are 2 compounds that we've published before, and you can see in the closed squares that these compounds cause dose-dependent reversal of A beta effects. But when these compounds are incubated in the presence of a constant concentration of the inactive sigma-2 ligand, the dose response curve is shifted to the right due to a competitive inhibition at the sigma-2 receptor. So I just want to remind you that these are all compounds that compete to the DTG binding site. You're going to hear more from this if you listen to Susan Catalano's talk, and if you read our paper that's been recently accepted for publication on a clinical trial candidate, CT1812. And this is similar to, in effect, to other sigma-2 compounds that we have tested from our proprietary compound, in that in vitro and in vivo and ex vivo, these compounds cause displacement of A beta oligomers, in addition to the effects on membrane trafficking. And you can see here, they end up causing a rightward shift in the potency of A beta oligomers in binding to the cells. What's the nature of this displacement? Well, we characterize this effect as a negative allosteric modulation of A beta oligomer binding. And the way we've demonstrated that is in a collaboration with Tara Spires-Jones and Christopher Henstridge, where we took frozen slices of human postmortem AD cortex tissue, and these were labeled for dense core plaques and for A beta oligomers by immunofluorescence. And then serial slices of this tissue was incubated with increasing concentrations of CT1812 in saline at 37 degrees for 1 hour. So it's important to point out that these are non-competitive conditions. We're looking at the endogenous oligomer bound to their receptors on this tissue. And what happens is that you can see displacement of A beta oligomer into the supernatant, increasing with concentrations of CT1812 as measured by ELISA and on Western blots. And if you look at the tissue, you see a decrease in the binding of A beta oligomer in a 2-micron halo around the dense core plaques compared to vehicle. And you see a bit of a trend to lower binding inside the plaque, but that was not significant. So this displacement by A beta oligomers by this sigma-2 ligand under non-equilibrium conditions can only be explained by an increase of the off rate and not by competition between the drug and A beta oligomers at the same binding site. So what is in this sigma-2 macromolecular complex besides TMEM97 and PGRMC1? I'm going to focus on A beta oligomer binding proteins, which have been most nicely reviewed by Steve Strittmatter and co-authors last year, a couple of years ago, actually. But there are a number of other proteins that associate directly with PGRMC1, that associate with TMEM97, and that associate with TMEM97, PGRMC1 together, which you will hear about if you listen to Aladdin Riyad's talk in this symposium. Again, one has to ask, do all these complexes respond to sigma-2 ligands? In a lot of cases, that's not known. And do the same sigma-2 ligands have the same functional effect in all of these interactions, or is it context dependent? We've used a proximity ligation assay to look at what proteins are involved in this macromolecular complex. This technique looks at immunolocalization of two proteins using primary antibodies and then secondary antibodies that have a DNA attachment to them, which form a template for amplification of a fluorescent signal. We can look at the detection of these PLA signals located in neurites or in the soma of neurons, and we can run this with technical control. We have run this with technical controls where we leave out one or more of the antibodies in the interaction to make sure we're seeing a specific effect. I won't show you the technical controls in all of the examples just for time and space here. Last year, we showed you that we could detect TMEM97 PGRMC1 interaction in neurons. We see a big signal in the soma, but we also see signal along the neurites. We've done further studies looking at co-localization of these sigma-2 proteins, PGRMC1 and TMEM97, with a synaptic protein, synaptotagmin-1. And you can see in both of these cases, we can see a signal from the protein pair that is significantly different than the technical controls. And also, we've looked at a biological control where you see no signal when you look at a synaptic protein and a nuclear protein, PARP1. So we know we're not looking at spurious signals, or we're looking at a minimum of spurious signals. We've extended these studies to look at these sigma-2 receptor proteins with two of the main A-beta oligomer binding proteins that have been best characterized. So here you see pair B, which in humans is LILRB2, co-localizes with TMEM97 and PGRMC1, and also cellular pre-bound protein co-localizes with both these proteins. And here you can see the signal being significantly higher for the protein pair than with any of the three technical controls. Now, what we're looking at is in the context of a neuron, and particularly in the context of a neuron synapse in Alzheimer's disease, we have a macromolecular receptor complex that consists of an oligomer receptor complex consisting of these three, possibly more proteins, and then a sigma-2 receptor complex consisting of PGRMC1, TMEM97, possibly other proteins that haven't been characterized. In the presence of elevated concentrations of A-beta oligomers that bind to their receptors, there's a downstream synaptic loss and damage, and resulting in memory failure. However, when a sigma-2 ligand such as CT1812 that has a therapeutic phenotype binds to its binding site on TMEM97, there's a conformation shift that's transmitted to the A-beta complex and results in displacement of that A-beta oligomer, restoration of synapse, and rescue of memory. So this is a complex system, but as in conversations with Wayne Bowen, he pointed out that it's not dissimilar to something like the GABA receptor, where you have a heteromeric complex with numerous different binding sites for different classes of drugs, and that binding of drug to one subunit affects the binding of a different drug to a different subunit. So I think we need to think of this particular receptor in this context in the same way. So in conclusion, we've seen that sigma-2 ligands can have multiple functional phenotypes versus A-beta oligomers in neurons, that a subset of these are negative allosteric modulators of A-beta oligomers, and that in the context of neuronal synapses, that there's a functional complex that exists of this, sigma-2 protein and an A-beta oligomer complex. What we really think is that data is needed on the functional overlap of compounds that interact with this system in this context with some of the other functional proteins, and hopefully we'll all look at the same compounds and compare the pharmacology and look more closely at the molecular entities that are involved. I'd like to thank all the folks at Cognition who have contributed to these studies, along with our academic collaborators and advisors, and especially like to thank Aileen for helping coordinate these presentations and to the Society of Neuroscience. Thank you very much for tuning in. I hope you listen to all the other talks in this seminar and join us for our panel discussions. Thank you.